So it's my pleasure this morning, afternoon, wherever you are, to introduce Dr. Nina Miolane. She's an assistant professor of AI at UC Santa Barbara in the Department of Electrical and Computer Engineering. She's also an affiliate researcher for the bioimaging at Stanford DISLAC. She's also co-founder and co-director of the Real AI Initiative at UCSB, whose mission is to ensure accuracy, robustness, fairness, and ethics for the deployment of AI to the applied sciences, of course, starting with brain sciences. She leads a research lab of about 15 members who advance multi-scale, reliable, deep phenotyping AI techniques to unravel the mysteries of the human brain, together with its normal and pathological variations. Her lab's research develops dynamic women-specific brain atlases, or these so-called digital twins of women's brains. She also creates early detection tools for medical conditions that predominantly affect women, like Alzheimer's disease and other forms of dementia. She ensures reliability and fairness of AI techniques and ensures accurate predictions across distinct subpopulation demographics. She has 10 years of experience in AI for healthcare across academic and industrial sectors, as in on the scientific advisory board for local companies at the intersection of AI imaging and healthcare. She was decorated with awards such as the L'Oréal UNESCO for Women in Science Award. It's celebrated as France's #1 woman's computer scientist by the president of the French Academy of Science. She also has an NSF Career Award and an NHR1. And she also, this is so cool, won the C3AI COVID-19 Grand Challenge, the $100,000 first prize. So that's pretty exciting. And of course, last but not least, she's also the co-founder and co-director, together with myself, of the AI Center for Excellence within the A&S Bowers Women's Brain Health Initiative, which was created last year. And of course, our goals at this point, hopefully you all know, but our goals are to harness the power of AI to accelerate reliable discoveries in women's brain health research and care. So please take it away, Nina.
Thank you very much, Amy, for the very kind introduction. And also, thank you all for putting this seminar series together, Sharon, Emily, Amy, and all the others. The previous talks were amazing, so I hope I'll do the series justice today with my talk. I'm very grateful to be talking and sharing our research today. I'm going to introduce the topic of our research, which is digital twins and the role of AI, artificial intelligence, in advancing women's brain health. And I actually like to start by addressing the simple question: why do we need to research women's brain health? Why do we, in fact, need a specific discipline that studies the brains of women? Women's brain health research is here to investigate conditions that uniquely affect women. For example, to answer questions such as, what is the effect of menopause on women's brain? Why do some women experience brain fog and why others do not? Women's brain health is also here to investigate conditions that disproportionately affect women and to answer the question, why do women have Alzheimer's more often than men? And this is a question that we've discussed in length the last iteration of this seminar series with Rachel Buckley, who talked about the sex difference in rates of mortality for Alzheimer's disease. And lastly, women's brain health research investigates conditions that differently affect women to answer questions such as, why does dementia present differently in women compared to men? And on that, I'd like to refer you to the fascinating research of Caitlin Casaleto at UCSF, which studies sex differences in dementia for women and men. And actually, it's very interesting because beyond the fact that women are disproportionately affected by dementia, they form 2/3 of Alzheimer's patients, they also present very differently, as in they are more resilient for longest period of time, so they don't exhibit symptoms, even though they have all of the signs of the pathology. But as soon as they start exhibiting symptoms, they decline more rapidly. So we need a dedicated discipline called women's brain health to investigate these conditions that uniquely affect women, disproportionately affect women, or differently affect women. Now, the problem is that this research has been historically not conducted. Despite the importance of these research questions that I just mentioned, they still have no answer. And this is because for far too long, scientific and biomedical research has excluded women and undervalued the study of women's health and women's brain health. And the resulting research gaps mean that we know far too little about women's health across the lifespan. And in fact, these gaps are even more prominent for women of color, older women, and women with disability. To illustrate that heartbreaking fact, let me share with you this groundbreaking study by Caitlin Taylor, Laura Priskett, and Emily Jacobs from UCSB. In their papers referenced below, titled The Scientific Body of Knowledge, Whose Body Does It Serve?, they have performed a historical review of papers published in neuroimaging. Indeed, when the MRI entered the field and became mainstream in the 90s, neuroscientists got an unprecedented peek inside people's brains. And they started investigating how brains were working when they were working well and how they were not working when they were undergoing some pathology. And we have, as they showed, thousands and thousands, in fact, tens of thousands of articles that have been published in neuroimaging, which is a significant boom for neuroscience studies. The problem is that women's brain health has not kept pace. In fact, across all of these over 50,000 neuroimaging studies, less than 1% focused on women's brain health factors. As the author states, studies on women's brain health factors amount to only 0.5% of the brain imaging literature. And on this graph, you can actually see the proportion of neuroimaging studies that look at women's health factors. They are represented by this tiny, tiny black bar at the bottom. So these are all the neuroimaging studies that talk about pregnancy, menopause, menstrual cycle, hormone therapy, and so on and so forth. We do not know enough about these very important research questions. And this is not just a heartbreaking fact. Actually, this lack of research has impacted women in ways that we are just starting to understand. For example, last year in 2023, you might have found headlines about the latest approved drug for Alzheimer's disease, the Leqembi drug. And this drug, in fact, only slows the cognitive decline of Alzheimer's disease. I mean, it slows the cognitive decline of Alzheimer's disease mostly in men. This drug slows cognitive decline in women by only 12% compared to 43% in men. So the lack of research has already had dramatic consequences on the way we understand women's brain health, but also on the way we treat women. Now, there are reasons to be optimistic. And actually, last year in November 2023, two big reasons gave us hope. November 2023, women's health started gaining significant traction. First, we got the launch of the White House Initiative on Women's Brain Health on November 13. President Biden announced the first ever White House Initiative on women's health. And this was followed by concrete actions a few months later, where he signed an executive order and also called on Congress to make an investment of $12 billion in new funding for women's brain health. In November 2023, women's health got traction also in California in another way, where on November 16, so actually a few days after the announcement by the White House, the Ann S. Bowers Women's Brain Health Initiative launched in San Jose to create the world's largest database dedicated to women's brain health. And the director of this initiative is Emily Jacobs, who is a co-organizer of this seminar series, hosted actually by the Bowers Women's Brain Health Initiative. So women's health started to make the headlines last year in November 2023. It started getting attention and started getting more funding. Meanwhile, November 2023 was also an important date in the field of AI, because it marked the anniversary of ChatGPT, which is an AI, an artificial intelligence, that can answer questions by predicting the next word in the sentence. So for example, if you asked ChatGPT or the latest model called ChatGPT-4, what is Alzheimer's disease, you will get an answer. Alzheimer's disease is a progressive neurological disorder that affects memory, thinking, and behavior, and so on and so forth. And ChatGPT in November 2022 took the world by storm by really surprising different users in the way it could predict text so easily. The way ChatGPT works is by predicting the next word in a sentence. It will read my question, which was, what is Alzheimer's disease, and give an answer by predicting next words. AI didn't stop there in terms of making the headlines, because a year later, ChatGPT had a video counterpart called Sora. And this is very similar to ChatGPT, except that it works on predicting the next frame in a sequence. So let me play the video. This video is entirely AI generated. It shows some archaeologists uncovering interesting objects on the archaeological sites. So you may note that there are a lot of physical inconsistency in this video, including a guy who just disappeared behind another guy. But it's still very impressive how convincing the video is. And similarly to ChatGPT, it's about predicting the next item in a sequence of items. While ChatGPT was predicting the next word, Sora predicts the next frame in a sequence. So following all of this news in November 2023, on the one hand, great announcements regarding women's health. On the other hand, even more announcements regarding AI, one cannot help but putting the two together and wonder if AI has a role in play in advancing women's brain health. And looking at what we just saw with ChatGPT predicting the next word and Sora predicting the next frame, I would like to ask the question, what if an AI could predict the next brain? What if an AI could predict a brain evolution through time? With ChatGPT, I asked a question, it gives the answer by predicting words. Could I give an AI a patient's brain, and the AI would predict how the brain evolves through time? That is the idea behind what we call the brain digital twins. Digital twins is a concept that has been introduced in AI. You can find digital twins of a lot of different systems and processes. For example, digital twins of planes, of cars. But the concept has also entered the medical field, and I'm going to define it in the context of brain. So a brain, digital twin, is a digital model of a physical brain that serves as its effectively indistinguishable digital counterpart for practical purposes, such as treatment planning. So for example, let's say I image my brain that will be represented by the left part of this logo. Then the digital twin will create an entirely digital model of my brain that should react exactly as my brain, should it be modified slightly. So for example, if I wanted to know whether I need to take this or that treatment for a given disease, instead of taking the treatment myself, we would first give the treatment to the digital twin, see how that digital twin reacts, and based on that, either go with a go or no go for me to use the treatment. That will be interesting, for example, for birth control pills, because there are a lot of birth control pills out there, and different people react differently to different birth control pills. So what if we could have a digital twin of a given woman, try the different pills in the digital twin, see how the twin reacts before effectively giving them to the person. instead of just experimenting on ourselves. So that's the vision, that's the dream for the brain digital twins. And today I'd like to ask the question, how could these brain digital twins really help us advance women's brain health today? So this will be the outline of the talk, titled again, Digital Twins and the Role of AI in Advancing Women's Brain Health. First, I'm gonna do a brief history of digital twins, but actually of digital twins and two related concepts called the atlases and the templates. So that's gonna be part one, a brief history of brain atlases, templates, and digital twin. In a second part, we'll see how we can start building women-specific digital twins. Why do we even need them? And how they can look like. And third, we'll go back to AI and answer the question, what is the role that AI can play in building these digital twins? Okay, starting with a brief history of brain atlases, brain templates, and brain digital twins. Because these are actually three closely related concepts, the digital twin being the most recent ones. And I'd like to give the proper definition so that we know what we're talking about. So first, the brain atlas. A brain atlas is a neurological map that defines discrete brain regions label. These labels are discrete, so that's why they are represented with a discrete set of colors on this one example of brain atlas called the Brain Tome from 2016. The brain atlas, in that sense, with these discrete brain regions that form a parcellation of the brain into anatomically important regions, is different from the brain template, which is a bit more continuous in nature. The brain template looks like a brain image, except it's kind of an average of brain images. It is computed by a given method to represent the average typical brain. So on this image in the center, you can see the most common brain template called MNI 152, which may look fuzzy. If you look closely at inside this image, you see that the circumvolution at the surface of the brain look a bit fuzzy, but that's because this brain is actually a model. It's an average of the brains of 152 subjects. And lastly, in this series of definition, we have the brain digital twin. The brain digital twin, by comparison with the brain template, is personalized. It is meant to be a replica of one given brain. For example, my brain. It's not the average of 152 brains. It is the replica of my brain, or one brain. And furthermore, we can experiment with it. So while the brain template is a mostly static image of a brain, the brain digital twin is a full model that we can use to test hypotheses. For example, I could vary this cursor here, which is the level of drug that I want to give to a patient. For example, the birth control pill that I'm thinking of giving to someone. And the digital twin is a full model that should react in response to me varying this drug. So these are really the main differences between brain atlas, brain template, and the brain digital twin, which is a full-blown personalized model of one person's brain. Now, researchers have been interested in these atlases, templates, and more recently, digital twins. So I'm going to give a brief history of what researchers have done in this field. At the beginning, there were mostly atlases and templates. Again, the digital twin is the most recent addition. And as soon as in the 1600s, we had the first atlases and templates. These ones were drawn by hand from direct observation. Indeed, the human anatomy has been studied for many years with the motivating assumption that the better our anatomical knowledge, excuse me, the better our medicine. And so in the 16th century, direct observation of the interior of the brain were already performed by dissection. And then the doctors at the time will build the knowledge of the human brain anatomy, for example, the healthy brain anatomy, by mentally averaging all the brains they would observe by dissection. And this led to these beautiful drawings of the standard brain anatomy that we can call a template healthy brain anatomy. So this is what you see on the right here, drawing by Willis from the 1664. But then, more recently, we got access to medical images. And today, atlases and templates are automatically computed from brain images. So with the advent of magnetic resonance imaging, and the advent of highly performant computers, the mental averaging that the doctors were doing in the 1600s got replaced by a computer average of the brain anatomy. So this is what you see on the right. This is a brain template computed from the OASIS database, entirely automatic. This template is typically an average of the brain images. Okay, so now we can compute brain templates automatically. What goal do they serve? A brain template serves two main goals. First, it represents the state of knowledge about the human brain. So we can see that the typical human brain, as shown in this template, has a series of circumvolution called gyri and sulci. And the distribution of this circumvolution on the surface of the brain is representative of what we know about the brain. So it has a goal of sharing the state of knowledge about the human brain. The second goal is for the brain template is to be used as a reference to align subject's brain in any neuroscience analysis. So let's imagine we have this brain template and a new patient comes in. We would like to know whether this new patient has a condition. What we're gonna do is we're gonna compare the patient's brain to the brain template and ask the question, how much does this patient's brain deviate from the brain template? With the assumption that if the deviation is too much, then this might be indicative of a disease. Which is that to answer the question, statistically, is the deviation that we observe indicative of a disease like Alzheimer's disease? So the brain template really serves two crucial goals. Now, let's look at brain templates that have been computed by researchers. And we're going to see that the structural brain templates are actually mostly built from men subjects. So this is the maybe 8 most common brain templates. And I've indicated in these 2 lines the number of men and the number of women that have been, whose images have been used to compute the brain templates. So the very first brain template of 1998 was actually built from the brain of one woman. It was a 60-year-old French lady. But after that, all the brain templates that have been built predominantly from men subjects. And actually, if you sum all of these data, you can see that the most common templates have been computed by averaging the anatomies of 432 male images versus only 162 images of female. And this is true for the MNI 152, which was first computed in 2001 and updated many times since then. But this brain template uses 86 men and only 66 women. Now, remember, the brain template is the starting point of every analysis. This is what we compare our patients' brains to. So there is a case in saying that this is biased towards men's anatomy. Now, population-specific atlases and templates have started to emerge. We have ethnicity-specific brain templates. For example, there is an atlas of Chinese adults that contains 2020 subjects with age spanning from 20 to 75 years. We have age-specific brain templates, for example, baby brain templates, developed for subjects younger than 12 months old have been developed, and they are reviewed in this paper by Oishi et al. However, there is no women-specific brain template. And that's an interesting gap, given that, as we saw, the women's brain present differently to neurological diseases. The women's brain present conditions that are only affecting women. So it's interesting that we have, as of yet, no women-specific brain template. It's interesting. Is it a problem too? Well, there are pitfalls of these biased brain templates. And this was actually one of the topics of my PhD thesis. The templates, remember, averages brain anatomy. If the variability of the brain is too large in a given region, then the computation of the template has two choices. Either the template will look very, very fuzzy because it's going to try to average different structures that vary a lot, or we will try to force a good resolution for this template. And in that case, the template will choose to represent one anatomy versus another. So imagine trying to take an average of two brains, while one has 2 circumvolution of the surface, and the second one has 3 circumvolution of the surface on its surface. What should the template show? Either it's gonna choose to not show anything and just have a fuzzy, blurry brain surface, or it's gonna have to choose between showing either 2 circumvolution or 3 circumvolution. In both cases, we are not very faithful to the variability that exists in these brain shapes. And very often, we have a mix of the two. And when it's the second case, every subsequent analysis will be biased towards the anatomy that the brain template has chosen to represent. If it chose to represent 2 circumvolution, then all of the patients which have 3 will show large deviation compared to the healthy brain template. And that's going to bias the analysis. Conversely, all the patients which has 2 will show less deviation, and maybe we're going to miss some important differences. On this illustration, you saw this bias computed on a brain template from the OASIS database. And we see when it becomes orange or red, these are the region of the brain where the brain template didn't really know what to show. Either showed something fuzzy or had to choose between showing a certain number of circumvolution. And we see in particular in the center here, which correspond to the hippocampus, that this is a region where the brain template didn't know what to show, because this is a region that shows high variability across the population. So if we want to avoid all of these biases, we need specific brain templates. And a fortiori, we need women-specific brain digital twins. We need women-specific brain templates first, especially in regions that have high variability compared to men, but also within women across the lifespan. And for brain digital twins specifically, this need is even more pressing. Because remember that the digital twin is not only a static image representing the standard brain, it's a full model that is supposed to react as the true brain that it is designed to represent. If I vary a level of drug that I give to this digital twin, I want that brain to react the way its physical counterpart will react. So we need women-specific brain digital twins that are dynamic and personalized in nature, and that really represent how women's brain would react. Which leads me to the second part of this talk. How can we build women-specific digital twin? We're going to focus on one brain substructure to do that, the hippocampus, which is a highly variable brain structure, which is the one circled in red on this image. This is the one for which we saw a few slides ago that there was a high variability. So this is one for which we would like dedicated brain templates and brain digital twins, because it varies so much across people, across pathologies, across the lifespan, across men and women. The hippocampus is a brain structure responsible for memory, learning, and navigation. It changes in response to hormones. And it is also one of the first structures to degenerate in Alzheimer's disease, which, as we have seen, disproportionately and differently affects women. So this is for all of these reasons, this is a good substructure to start with in our quest to build the digital twin of women's brain. Going towards this digital twin, can we model how this hippocampus will respond to brain hormones? We have a brain digital twin. I want to be able to move that progesterone cursor and see whether the brain changes. Can we do this during the menstrual cycle? And can we do this during pregnancy? Let's start with building digital twins of women's brain during the menstrual cycle. So first, if we want to go towards a digital twin of the menstrual brain, we're going to need data. And for this, we are lucky because Laura Prichette has provided us with a high quality, densely sampled data set. She imaged her brain during her menstrual cycle. This is Laura Prichette. And for every day of one normal menstrual cycle, normally circling, she recorded her hormones level. So she recorded estradiol, progesterone, LH, and FSH, together with brain MRIs. So every day for the 28 days of her menstrual cycle, she recorded her hormone levels, and she went into the MRI scanner to take these brain images. This led to the creation of the data set called 28andMe, which is available on the open source platform OpenNeuro. The analysis of this data set showed that the hippocampus substructures were indeed changing in volume, confirming our hypothesis that they're highly variable, even for one person, Laura. So Katie Taylor took the data from Laura, segmented the different hippocampus substructures. So you can see here the hippocampus in the brain, and in different colors, the different substructures of the hippocampus. And she showed through volumetric analysis that different substructures of the hippocampus were indeed varying depending on progesterone. So on the horizontal axis here, you have progesterone values. And on the vertical axis, you have the volume of each of these substructures. And you can see that for the two substructures chosen here, they increase in volume as progesterone increases during the menstrual cycle, which is kind of crazy to think about because this is something that happens every month. Every month, progesterone spikes, and there is a resulting visible change on the brain shapes, and particularly on the shape of the hippocampus substructure. However, in this analysis, even though the different substructure of the hippocampus were shown to change in volume, there was no change observed in the volume of the total hippocampus. But this makes sense because a structure can change in shape without necessarily changing in volume. So even though each of the substructure were changing in volume, there were somehow balance changes such that the overall volume would not change, even though the shape was changing. And so we performed subsequent analysis to really show in 3D what was happening for this hippocampus. So this is a work performed by Adele Myers, where she extracted the structure of the full hippocampus, not subdivided into any substructures, and visualized in 3D how this hippocampus will change in shape during the menstrual cycle. So it looks like this. You're gonna see as progesterone peaks, the hippocampus changes in shape. Let me play that again. So it's really shearing and coming back to normal. This deformation indeed correlates with progesterone and presents as a shear. So I'm going to show this animation again, this time not with the day of the menstrual cycle being the varying variable, but here using progesterone as a cursor that is moved, and you'll see how that impacts the shape of the hippocampus. So you go here and you can see this shearing deformation. Do it again. Boom, the hippocampus will shear. So even though we knew that there were some volumetric changes happening in this different substructure, we didn't really know what was the overall shape change associated with progesterone. And thanks to this 3D analysis, now we can vary the progesterone cursor and see that the hippocampus is actually presenting a shear when progesterone increases during the menstrual cycle. So this is really the very first iteration of a digital twin of the menstrual brain in 3D and in reaction to hormone levels. But can we model how the hippocampus responds to brain hormones during another life-defining event, and in particular during pregnancy? So moving towards a digital twin of the pregnant brain, we can also seek to apply similar technique, but again, we will need data. And here we are again lucky because the professor Liz Chastil has imaged her brain during her pregnancy. So this is Liz. And during the course of her first pregnancy, starting 2 weeks before the start of the pregnancy and continuing several months after birth, Liz Chastil also recorded her hormones level and also went into the MRI scanner to image her brain. And that gave us an unprecedented insight into what happens in the woman's brain during pregnancy. Again, volumetric analysis were performed during pregnancy and after birth by Laura and Caitlin. And I'd like to note that the changes in hormones during pregnancy are so much higher than the changes in hormones during the menstrual cycle. So the little effect that we could observe on the digital twin of the menstrual brain are meant to be much higher, much stronger for the pregnant brain. Because you can see that, for example, progesterone is easily multiplied by a factor 100 during the gestation weeks. So on the right, you have the scale of progesterone. It has some values which correspond to the values of the menstrual cycle, but at the peak of the pregnancy, these values have been multiplied by a factor at least 100. And this is similar for estradiol, which is shown on the left, which also gets multiplied by a factor 100 to 1,000. So progesterone already had an effect on the hippocampus during the menstrual cycle. What kind of effect can it have on the brain during pregnancy while its level just skyrockets? And volumetric analysis showed that indeed this life-defining event that is pregnancy has an effect on the brain. On this chart, you can see how the total volume, the total gray matter volume decreases during the pregnancy stage to re-increase a little bit post-pregnancy, but not to the level it was before the pregnancy. So before the pregnancy, we have a total gray matter volume that is depicted in pink here, that decreases until birth and then re-increases slightly only after birth. This is volumetric analysis that kind of confirm our hypothesis that the brain is highly variable and therefore cannot be represented or should not be represented by a unique brain template. Can we image all of this and model all of this in 3D using a digital twin? So we trained an AI model on brain shapes to learn the deformation associated with hormonal variations. So here you're gonna see something that doesn't look like a brain because it's the hippocampus. It's a substructure of a brain. The different color correspond to the different substructures of the hippocampus. And on the right, you have sliders that you can move to change the hormones that are presented to this hippocampus during pregnancy. And you're gonna see as a result that we can observe with the eyes the shape variation of this brain structure. So this is our hippocampus that exists in 3D. Different colors represent different anatomical substructures. And on the right, we have the three main hormones that vary during pregnancy. So estrogen first can be moved, so we can see how this digital twin reacts when we change the hormonal levels. And we can do the same thing with progesterone and LH. Again, this is a model that has been computed from these terrestrial data, but everything you see here is a digital twin of a hippocampus. This is AI created. And we can use that digital twin to try to understand then the mechanistic reason of why this brain is undergoing these changes or not. So to wrap up on women-specific digital twins, the field is still in its infancy, but still, since we start to have the first densely sampled data set of women's brain during defining events such as menstrual cycle, pregnancy, we can start building 3D models of specific brain structures and their response to hormones levels. We are building all of that into an app that will be available online soon, that will be called Her Brain, and you will have access to these digital twins, and you will be able to play with the different cursors and see how women's brain react when hormones varies. Wrapping it up, I'm gonna enter my last section on the role of AI in building these digital twins. As I've said, digital twins are models that predict how a given brain, the brain of Laura, the brain of Liz, will change under different settings. For example, when we vary the hormone levels. These models are built using AI, artificial intelligence, meaning computational techniques that automatically learn patterns from brain images. AI learns patterns from brain images. They learn from data. And therefore, these AI models are only as good as the data they see, which leads us to one little problem when we try to use AI in building digital twin, is that the data sets are very small, very, very small, the data set of women's brain images. So to give you a feel of how small they are, let's compare women's brain data sets to traditional image data sets that are usually used to train AI models. The most famous imaging data set for AI is the so-called ImageNet. This has 14 million images. By comparison, general medical imaging datasets are already way smaller. One of the largest, called MedPix, has 60,000 images. That's already 200 times smaller than the datasets of images that AI is traditionally trained on. Now, women brain datasets are even smaller than that, a factor of 600 times smaller compared to traditional medical imaging datasets. And by women brain dataset, I don't only mean brain images of women. I mean brain images of women together with other variables that allows us to study women's health question. For example, a women brain MRI together with a hormone level, or women brain MRI together with the stage of the menstrual cycle. Data sets that were built to ask and answer questions on women's health. These data sets are rare, and when they exist, they are incredibly small. And if you want to do the math, that's a factor 120,000 times smaller than the traditional data sets that have been used to develop all of AI. So even though AI is in a boom, there are over 500 AI research papers published every week. We need to keep in mind that these models were not built for this data regime, for the data regime that we need to analyze the current data sets that exist on women's brains. So we need to enhance or generalize AI models so that they can learn more from less. And that is actually the methodological research that is performed in our lab at UCSB, which is called the Geometric Intelligence Lab. And the whole idea behind geometric intelligence is to first meaning before applying an AI model, we first try to understand what are the characteristics of the data, what is the characteristic of the space of the data, in order to restrict what AI has to learn. We seek to build in as much inductive bias as we can before we call the AI model. Giving you an example on the digital twin that we just saw. Consider the data space of brain anatomies. So it's an abstract data space in which each data point represents a whole brain. So for example, this green data point in this data space represented by this curved black space, this green data point represents a whole brain. Now, not every shape can be a brain shape, which means that the data space of brain anatomies is highly constrained. It's actually equipped with a lot of complicated nonlinear constraints. These constraints turn the data space into a nonlinear space, which is why this grid here is showed as curved. It's what we call a nonlinear manifold. Now, it's interesting to take into account this nonlinear manifold because by considering the constraints that apply to our brains, we can significantly reduce the dimension of the data space and therefore reduce the number of data points that we need to train an AI model. So the goal in our lab is to generalize existing AI methods from their traditional presentation on linear spaces to the nonlinear manifolds. And we have a lot of work because even if we look at the most simple AI model or machine learning model, like linear regression, fitting a line through data point, this was a model that was developed in the 1800s. The corresponding model for these curved data spaces actually only came way later in the years 2000, 2011. So if linear regression took 200 years to be generalized, you can imagine that the latest AI models also need to generalize. So now what's next? I've presented digital twins, but mostly in the context of structural digital twins of brains. So we looked mostly at the anatomy and brain MRIs. But moving forwards, we're gonna need multi-scale and multimodal digital twins. Multi-scale meaning that the brain is a multi-scale system. it's not only a macroscopic scale system, not only these brain MRIs that we can see at the macroscopic scale, but it also has different processes that happens at the nanoscopic scale, microscopic scale, eventually leading to the behavioral symptoms that we can see. And it's also highly multimodal. There are a lot of different variables or data types that we can record to study a brain. So as mentioned, I mostly presented studies using the brain MRIs, but we could also record the electrical activity of neurons. We could recall cognitive measurement to really pick a more complete picture of this brain digital twin. And by doing this, we're going to be able to fight this lack of data in AI by incorporating diverse data sets, some with missing data, some very noisy, some across scale. We can hope to have this digital twin that provide not only a better understanding of the brains, but also potential better diagnosis accuracy when it comes to predicting the evolution of diseases. And that's something that we've been very interested in my lab, where we have developed this method called Bayesian hierarchical network for combining data sources in medical diagnosis. Now, we haven't applied it yet to brain images, but we did apply it to COVID back in 2020, where we participated in the C3AI Grand COVID-19 Challenge. And we showed that by incorporating these different data sources, we were indeed able to improve the diagnosis accuracy of the worst at-home testing kit from 68% to 87%. An AI model that gave us the first prize at the C3AI COVID Challenge. So wrapping up, I've introduced the concepts of digital twins and the role of AI in advancing women's brain health. I've presented a brief history of these three related concepts: brain atlases, brain templates, and digital twin. I showed you initial steps that we can take in building women-specific digital twins, and specifically for the hippocampus substructure of the brain. And lastly, I've concluded on how AI can help in building digital twins if we are able to generalize AI models to the really low data set sizes that we have in women's brain health. I'd like to thank the Geometric Intelligence Lab at UCSB, a lot of really good PhD students and postdocs, and of course the Women's Brain Health Initiative, which has really gathered very smart and important people to advance this field all together. I thank you very much for your attention. I'll be happy to take questions.
I'll clap for everybody. That was a fantastic talk, Nina. Thank you so much. All right, so we have a question. Thank you for a great inspiring talk. I'm a newbie here with a very newbie question. To get complete and useful data sets, how are hormone levels measured alongside brain imaging?
Yeah, thank you for this question. So for the two data sets I referenced, the first one being the 28andMe data set of the menstrual brain evolving through time by Laura Priskett, Caitlin Taylor, and Emily Jacobs. Hormones levels were measured by blood sampling. And actually, same thing for the second data set that I mentioned, which is the one by Elizabeth Frastil, with also collaboration with Laura, Caitlin, and Emily Jacobs. Same thing, blood samples.
Great.
Which is indeed more accurate than other measurement modalities like urine or, I don't know, sweat or other modalities. That's why they wanted to have this capability.
This next question is one I had too. How many months after delivery was the post-brain volume measurements? Did they check whether it returns to original volume after a while? Otherwise, it might be a problem for women who have 5 or 6 pregnancies or more. So.
Yeah, I think it was, let's see.
2 years postpartum.
Exactly.
And Magdalena Martinez Garcia, who's now a postdoc with us here at UCSB, has done some work And I should just say, this is a plug for the next talk with Professor Susanna Carmona, because she's going to go over this whole sphere of the maternal brain and how it changes across pregnancy. So maybe just stay tuned for that. But Magdalena finds evidence that these sort of changes are evident up to about 6 years postpartum, likely far beyond that. So these seem to be kind of like permanent etchings in the brain. And I will just push back that I don't think that that's necessarily a problem, but stay tuned for next week to find out from the people that are really leading these scientific questions.
Yeah, and there was actually just a
I'm in.
recent UK Biobank study predicting number of children, or basically if you've had children or not, from the T1 anatomy, and they actually used a brain age model in between that. So they actually showed that people who had lower predicted brain ages for their chronological age had children. So it seems like whatever brain age model they're using is predicting a lower brain age for people who are parents. So that was an interesting recent one in about 30,000 subjects. So it's always good to see these larger end studies.
Do you know how it compounds with several pregnancies? Because I think that was part of the
Yeah,
question.
that at least the UK Biobank one, they could predict whether you had children or not. But as far as like the number of children, it was very difficult to do. So I'm not sure if there's much signal there, at least in the UK Biobank data set.
Yeah, maybe a first drop for the first pregnancy and then stabilize it. Yeah.
Exactly. Yep. Yep. Good. So I just had a brief one. We have like a couple more minutes. Maybe for those people who don't work with digital twins or AI, could you give like a couple of really concrete examples of how digital twins could help in like clinical management of women's health conditions?
Yes. So the whole idea of the digital twin is that it's a personalized model of someone's brain. So for example, we built one digital twin of Liz Trastill's brain. And the idea is that it's not just a static image that represents Liz's brain anatomy. It's really a model that we can poke to test hypotheses. So for example, I may want to know how Liz may react to a given treatment instead of just taking the results of some study that has shown that on average, this treatment worked for this amount of women. Or instead of giving that treatment to Liz directly, I could give that treatment to Liz's digital twin and see how the digital twin react. And we're not there yet, but with the next generation digital twin, hopefully we'll get an answer of the type, yes, this is a good drug to treat this condition, or no, that will not have any effect on the brain like Liz, and therefore something else should be tried. So a concrete application will be, instead of testing drugs on the actual people, test drugs on the digital twin, both for the treatment, but also for a clinical trial. Clinical trials are very expensive, both in time and money. If you could just use the digital twin to get some ideas of what might or might not work, we could at least orient the design of the clinical trials and only test the most promising option in humans.
Nice. Okay. We have a question from Andrea. Great talk. I'm wondering whether clinical data could be leveraged to increase the size of the data, and if so, what are the barriers to overcome?
Yeah, thank you. Great question. So clinical data,
like a clinical MRI, which is actually, I think, what Emily and you all are doing in the kind of UC system, right? You're kind of collecting these MRIs that are generally being done, I think, clinically. Is that correct, Emily?
So to start, these are at research dedicated.
Oh, okay. They're research studies for now.
But we will have a planned expansion to include, clinical scans.
Yeah. So that's definitely something that could be done, but there's always, IRB issues and things like that with clinical imaging, and then it's harder to share that. And yeah, anyway, but yeah, that could be potential candidate for
exactly. I mean, the more data we can gather, the better. But if it's data that have been collected in a clinical setting, there might be privacy issues that are hard to go around. Which is why the data we use right now have been collected, as Emily said, in the context of research purposes. Yeah.
Okay, we'll take one more question, I think. This is kind of a mashing of the two remaining questions. Would digital twins therefore only be able to work by collecting a lot of data in a specific person, for example, 30 days of consecutive imaging plus hormones, in order to predict how a person will react to a specific pill? How realistic is this for clinical interventions?
Only work by collecting a lot of data on a specific person. So that's what we started to do. And again, this is really the first iteration on this concept of digital twin for women's brains. But the goal is that in the future, you don't need to collect as many data on one woman in order to have a digital twin. Because you would have some women that have been densely sampled. For example, Liz has been sampled every 2 weeks during her pregnancy. And then you will have other women that have been sampled maybe only once, but I will be able to showcase the diversity of the population. And so by using a mix of the two, when a new woman comes into the study, you would basically match her to the women in the database that is the closest in phenotype. And then maybe these other women in the database had been sampled densely and can be used as a baseline to understand how the new women will react to a drug. So hopefully in the future, we'll have a database that's large enough to include largely densely sampled women, and then also a wide variety of different women to build personalized digital twin of a new woman coming to the study.
Yeah, and just to add to that, there's a lot of exciting work in transfer learning and sort of ensemble approaches with small data. So we did this study where we showed you could kind of use a big densely sampled data set to predict what would happen in a new individual if you just had a few samples from them. So yeah, this is the idea.
Good. Yeah, exactly.
Okay, let's stop it there. Thank you so much, Nina.
Thank you very much.
And we'll see you next month. Yep. Look for
Sorry, maybe I can share the.
Yeah. Perfect. Yeah, so for those of you interested in pregnancy and postpartum and parturition, this will be perfect for you. All right, thank you so much.